Welcome back, everyone. So we are in part 5 of our topic 4, genetic blueprint and energy production. So in the last video, we learned about the first step of our process of cellular respiration. So now we're going to use the products of glycolysis and take it into the Krebs cycle and continue breaking down our 3-carbon chain pyruvate, extract more energy, and continue on to the electron transport chain. So by the end of this video, you should be able to describe the relationships of glycolysis, the Krebs cycle, and the electron transport chain. You should also be able to identify the amount of NADH, FADH2, and ATP produced during cellular respiration. And then finally, you should be able to state the overall chemical equation or formula for cellular respiration. Okay, so before we get into the Krebs cycle, let's just review where we are. Okay, so we have discussed glycolysis in the last video, where we took glucose and we broke it apart, right? So we broke it into 2 3-carbon pyruvate molecules. So that pyruvate is now going to enter the mitochondrium. So during this process, the rest of the parts of cellular respiration are occurring within the mitochondrium of the cell. Now notice we have not yet gotten into the oxygen part. Okay, so we won't get into that part until the electron transport chain part. Okay, and so far the only thing we've done is we've broken glucose into 2. So we haven't produced any carbon dioxide or water yet. Now, before we get into the Krebs cycle, we have to have this little transition step that takes place. So this is just to kind of prepare the pyruvic acid to get into the Krebs cycle. So remember, we have this 3-carbon pyruvic or pyruvate, or otherwise known as pyruvic acid. It's the same thing, that gets transported from the cytoplasm into the mitochondrium, specifically the matrix of the mitochondrium. And when it's there, it goes through this little transition step. And this transition step involves basically a coenzyme. So a coenzyme is basically just this thing that attaches to a molecule, but it doesn't really change the molecule in any way. Okay, so it's not really, it just kind of attaches to it, but doesn't change the rest of the molecule. So pyruvate will undergo a reaction where it will lose a carbon, and by doing so it loses carbon dioxide. So one of the carbons gets released as carbon dioxide. Okay, so we lose a carbon dioxide. And by losing a carbon dioxide, we strip some electrons from that process and add it to an electron carrier. So we have that NAD plus then gets reduced to NADH. Okay. And then we have this coenzyme that gets added in. So now our 2-carbon chain with the coenzyme A added becomes something called acetyl-CoA. So we have acetyl-CoA is the end product that goes into the Krebs cycle. Okay. So this happens twice, right? Because we have 2 pyruvate molecules. So at the end, we end up with 2 acetyl-CoA molecules entering the Krebs cycle. So here's our summary. Our inputs, we have 2 3-carbon pyruvates. So our outputs, we end up with 2 2-carbon acetyl-CoAs. We add a coenzyme. Okay, so that coenzyme gets added to the 2-carbon molecule. Since we lost a carbon, so that pyruvate gets broken down. So that loss gets outputted as carbon dioxide. So since we lose one carbon from each of the pyruvates, we end up with 2 carbon dioxides being released. Okay? And that energy or the electrons that get stripped in the process get added to our NAD plus to form NADH. So we have 2 NADH from that process. So this is the outputs of our transition step. So now we're going to take that 2-carbon acetyl-CoA into the Krebs cycle. All right, so let's talk about the Krebs cycle. Now you'll see in this diagram here, which came from your textbook, it doesn't say Krebs cycle, it says citric acid cycle. So I don't know why your textbook says Krebs cycle, because most textbooks these days call it the citric acid cycle. The reason why you'll see some textbooks call it the Krebs cycle, it's just named after one of the people that kind of put together the understanding of this process. So, there was a person who named Krebs, who basically was one of the scientists who discovered this cycle. He wasn't the only one, which is why he didn't even call it the Krebs cycle. He called it the citric acid cycle. So for some reason your workbook continues to call it the Krebs cycle, so that's why you'll see it in the workbook, but you will also see it sometimes as the citric acid cycle. So most textbooks these days call it the citric acid cycle. So if you see it like that, it just is the same thing. Krebs cycle, citric acid cycle, it's just interchanged. Okay, so instead of calling it by the guy's name, it's called by just a generic name. Okay. So this occurs in the matrix of the mitochondrium. So if we think of the mitochondrium, remember it looks like a little kidney bean. So it's kind of in the center part of that kidney bean. So within that jelly-like center. And this is where we have this kind of cycle, this loop. And notice it's a closed loop. So we end up adding something and then it just goes through. And then it'll keep, the process will keep going if there's something there, okay? And this total has 8 chemical reactions, and luckily you don't see them, right? So you don't have to learn them. And so biology majors, they have to learn them, and it's very tedious, all these chemical reactions that take place. But what you do need to know is what happens. So we have our acetyl-CoA that enters the citric acid cycle. But prior to entering, the coenzyme comes off. Okay, so there goes the coenzyme. The coenzyme just gets recycled. So now we just have the 2-carbon molecule enter the citric acid cycle. And as it enters the citric acid cycle, it actually binds to a 4-carbon molecule. That starts this whole one of the chemical reactions, okay? And as it's going through the citric acid cycle, it's undergoing these chemical reactions, and as a result of these chemical reactions, we end up getting some electrons stripped. So we have, for each of these acetyl-CoAs that go in, we end up getting 3 NADH electron carriers reduced. We get another electron carrier, okay, so FAD is the oxidized form, and then when it obtains the electrons from these chemical reactions, the reduced form is FADH2. So that's a couple more electrons get added to that, okay. We also produce 2 carbon dioxides, okay? So there goes the last of the glucose molecule, okay? So remember we started out with, our 6 carbon glucose. We lost 2 of the carbons for carbon dioxide during that transition step, and then we also lose 4 more, 2 here and then 2 for the other pyruvate. So there's our 6 carbons from our glucose molecule are now gone off. All right. And then in this process, we also produce some ATP. Okay, so for each of the acetyl-CoAs that entered, we produce 2 carbon dioxides, 1 ATP, 3 NADH, and 1 FADH2. These again being the electron carriers. So here's our summary. So we ended up having 2 of the 2 carbon acetyl-CoAs go into the Krebs cycle. Outputs, the coenzyme gets stripped off, right? The 2 carbons get released as carbon dioxide. Okay, actually that should be 4, right? Because it's 2 per each. So scratch that, that's a 4. We have we had at the beginning, we had our NAD+, which is the oxidized form. We had electrons added to that through those chemical reactions to get reduced to 6 NADH electron carriers. And then we had the FAD, another electron carrier, FADH2 produced. And then we have ATP being produced. So those are our outputs of the Krebs cycle. Okay. So from there, we've completely broken down our glucose. So now glucose is completely gone. The NADH and the FADH2 will head into the next part, the electron transport chain. Okay. So far, we've completely used up that glucose. Okay, so we haven't even gotten to the oxygen yet. And so far, we've produced 4 ATP total. So now we're going to take the carriers, the electrons from the carriers, NADH and FADH2 from the Krebs cycle and from glycolysis. Remember, we got some glycolysis. And those carriers will enter the electron transport chain where we will finally use that oxygen. So let's talk about the electron transport chain. So remember all those electrons that were carried by the NADH and the FADH2? Well, those will get stripped off and they will be passed through this complex of inner membrane proteins, these integral proteins. And so you can see this is what we call the inner membrane of the mitochondrium. And you can see these big proteins. So these are integral proteins. They're actually little protein channels, some of them, not all of them. And these electrons, they get stripped off. So NADH will get oxidized to NAD+. Okay, same thing with FADH2 will get oxidized. Remember, oil, oxidation is losing, to FAD. Okay, so those were oxidized, and then they can go back into glycolysis, outside into the cytoplasm or into where the citric acid cycle is, Krebs cycle, to gain more electrons. Okay. So now these electrons are moving through the inner membrane, passing through these, these components of the inner membrane. And by doing so, they have just enough energy to them that it provides enough energy to pump what we call protons, or H+, across the inner membrane into what we call the intermembrane space. So we end up pumping all of these protons, or H+, across into this intermembrane space. And this creates an electrochemical gradient where we have all these positive protons on one side of the inner membrane. Okay. So think about that. Well, for now, just imagine a bunch of hydrogen ions, or H+ on this side. Now what about all those electrons? So now we have all these electrons passing through. So the final electron acceptor that's going to accept those electrons is oxygen. So remember that oxygen we talked about? So oxygen is going to be the final electron acceptor. So it's going to accept those electrons and bind with water, or with hydrogen to form water. Okay, so oxygen is the electron acceptor and the final one, and water is produced in that process. Okay, so that is the electron transport chain. So by the end of the electron transport chain, we end up with a bunch of water where all the electrons are attached, and so it's stable now. And then we have a bunch of hydrogens on one side of the membrane. Okay, so now we have to worry about all these hydrogens on one side. So next we go into something called chemoosmosis. So over here, okay, so just imagine somewhere nearby there is this weird shape protein which has it's basically an enzyme, and it has a weird shape to it because it has a little, like a generator, a little rotor in it. And what happens is when it opens up, all of the hydrogens on this side of the inner membrane space will diffuse across, okay, because it's going from high concentration to low concentration. So as it's diffusing across, it's going to cause that little generator to rotate, and the flow is going to cause again that generator to rotate, producing enough energy to convert ADP into ATP. So we call this entire process chemoosmosis. Okay? And this process pays off big. It produces anywhere from 30 to 32 ATP on average. That's a lot. Okay, so this electron transport chain pumps so much hydrogen across the membrane that when that channel opens, with that ATP synthase is working, we get a big payout of anywhere from 30 to 32 ATP. Okay, so that's where the bulk of the ATP is produced during cellular respiration. Okay, so here's our summary of the process. So our inputs, remember, we have our electron carriers, so including the ones from glycolysis and from the Krebs cycle, citric acid cycle, and the transition step, we had 10 NADH. So those will get oxidized to 10 NAD+. Okay, we have 2 FADH2s that we got from the Krebs cycle, citric acid cycle. They will get oxidized to FAD. We brought in 6 oxygen molecules, and those will become water. Okay, and then we had some ADP and phosphate ions hanging around, and we produce ATP from that. Okay, so this is the outputs of our electron transport chain. So if we look at our summary here is the summary of the entire equation. So we start out with our glucose. Remember, our glucose is that 6-carbon molecule. We break down the carbon first into pyruvate, which is that 3-carbon chain molecule. We do that in glycolysis, and then that goes into the transition step, and then further into the Krebs cycle, where it gets broken down into carbon dioxide. Okay. During that process, we get some electrons added to our electron carriers. Okay, and so those electron carriers are going to be carrying electrons to our electron transport chains. We also gain some ATP from that process. Okay. And then once we get into the electron transport chain, those carriers drop off those electrons, which provides enough energy to pump hydrogen ions across the membrane. And once it's across the membrane, it creates that electrochemical gradient. So when that ATP synthase opens up, hydrogen ions diffuse across through chemoosmosis, which generates enough energy to yield a bunch of ATP. So we end up with the final electron acceptor being oxygen. So that's going to pick up all those electrons to form water. And then we end up with a bunch of ATP. Okay, so there's our overall equation of where everything was inputted and outputted. All right. Now, because this is such a complicated process, I always turn to the Neba sisters for help, so let's go ahead and watch a video.
Are you a morning person? One of us is, and one of us is definitely not. Mainly because when I wake up in the morning, it just takes a while for me to get my energy back. It takes a lot of time and coffee for that to happen for me. Cells really don't have that luxury. They are busy performing cell processes all the time. Active transport of many substances needed for their survival, for example. And the energy currency they need, specifically, is ATP. ATP stands for adenosine triphosphate. It's a type of nucleic acid, actually, and it is action-packed with 3 phosphates. We have a video all about ATP and how it works as an energy currency. So where am I going with this? Well, cells have to make this ATP. It doesn't really matter what kind of cell you are. Prokaryote or eukaryote, you have to make ATP. The process for making that ATP can be different, however, depending on that type of cell. One way is called aerobic cellular respiration. Lots of organisms can do aerobic cellular respiration, but this video is specifically going to go into aerobic cellular respiration in eukaryotic cells. That means this video is talking about the process within cells that have membrane-bound organelles, such as a nucleus and mitochondria. Eukaryotic cells include the cells you find in protists, fungi, animals, and plants. The mitochondria, which can be found in most eukaryotic cells, are going to be kind of a big deal in this aerobic cellular respiration because some of the process occurs in the mitochondria. So let's get started. Remember, the major goal for any organism performing this is to make ATP. Okay, here's the overall look at the equation for aerobic cellular respiration. Remember that reactants, inputs, are on the left side of the arrow, and products, outputs, are on the right side of the arrow. This equation, by the way, looks remarkably similar to photosynthesis. Look at how the reactants and products are on different sides. Well, that doesn't mean they're simply opposites. It does show that they have substances in common. In photosynthesis, organisms make glucose. Notice how glucose is a product. But in cellular respiration, organisms break the glucose down to make ATP. Fun fact, you know how when a bean seed first germinates in the ground, it isn't able to do photosynthesis yet? Yeah, the germinating bean is relying on glucose that it has stored, and it's doing cellular respiration to break it down to make ATP so it can grow. Of course, once it starts to mature and develop leaves, it can do photosynthesis. It will be able to do both photosynthesis and cellular respiration. Plants make glucose in photosynthesis, and they break it down in cellular respiration. They can do both. But if you aren't photosynthetic, such as a human or an amoeba, you have to find a food source to get your glucose. You need glucose to get this process started, and we're going to assume that we're starting with one glucose molecule. Step 1, glycolysis. This step takes place in the cytoplasm, and this step does not require oxygen. It's considered anaerobic. In glycolysis, glucose, the sugar from the equation, is converted into a more usable form called pyruvate. Glycolysis usually takes a little ATP itself to start up. The net yield from this step is approximately 2 pyruvate and 2 ATP molecules, and 2 NADH. What is NADH? NADH is a coenzyme, and it has the ability to transfer electrons, which will be very useful in making even more ATP later on. We'll get to that in a minute. Now an intermediate step occurs. The 2 pyruvate are transported by active transport into the mitochondria, specifically the mitochondrial matrix. In the mitochondria, pyruvate is oxidized. The 2 pyruvate are converted to 2-acetyl-CoA, which will be used by the next step. Carbon dioxide is released and 2 NADH are produced. Step 2, the Krebs cycle, also called the citric acid cycle, still in the mitochondrial matrix. The citric acid cycle is considered an aerobic process. While the cycle doesn't directly consume oxygen, some of the events in the cycle need oxygen to continue. To learn more about this intricate cycle where the 2-acetyl-CoA will enter, check out the further reading suggestions in the video details. Carbon dioxide is released. We also produce 2 ATP, 6 NADH, and 2 FADH. FADH is also a coenzyme, like NADH, and it will also assist in transferring electrons to make even more ATP. Step 3, the electron transport chain in chemiosmosis. This is just a beautiful thing, really. In eukaryotic cells, this is still in the mitochondria, and to be more specific, it's involving the inner mitochondrial membrane. We do require oxygen for this aerobic step. This is a very complex process, and we are greatly simplifying it by saying that electrons are transferred from the NADH and FADH to protein complexes in electron carriers. The electrons are used to generate a proton gradient, as protons are pumped across to the intermembrane space. All these protons being pumped out into this intermembrane space generates an electrical and chemical gradient. The thing is, if you remember from our cell transport video, ions, like the H+, don't easily travel across membranes directly without something to travel through. The protons can travel through an amazing enzyme called ATP synthase. If I could be any enzyme, I'd be ATP synthase because it has the ability to make ATP by adding a phosphate to ADP. ADP is a precursor to ATP. ADP has 2 phosphates, but if it obtains a 3rd phosphate, it becomes ATP. So, in chemiosmosis, the protons travel down their electrochemical gradient through a portion of the ATP synthase, powering it to make ATP, the ultimate goal. Oxygen is the final acceptor of the electrons. When oxygen combines with 2 hydrogens, you get H2O, water. If you remember from our equation, water is a Listic product. Now, the electron transport chain in chemiosmosis step makes a lot more ATP compared to the other 2 previous steps. How much? So I've noticed in my years of teaching and the assortment of textbooks I've collected over the years, there are some different numbers in cellular respiration charts. In fact, you can even see them change a bit in different editions of the same book. And it's made me want to emphasize that it's important to not just memorize a number, because it really is more of a range. I want to focus less on the number of ATP made per glucose molecule because it depends on a lot of variables. One variable is the gradient we were talking about. How many protons were pumped across the mitochondrial membrane? You can see more variables discussed in the factual references, and those references have estimates ranging from 26 to 34 molecules of ATP per glucose molecule in the electron transport chain in chemiosmosis step alone. Then, if you add the other 2 steps, Krebs, otherwise known as the citric acid cycle, and glycolysis, you can estimate a range of anywhere between 30 to 38 net ATP molecules total per molecule of glucose. Again, to emphasize a range. Now, this was just one way of creating ATP, but like we had said at the beginning, all cells have to make ATP, but the way they do it can differ. If there's no oxygen available, some cells have the ability to perform a process known as fermentation. It's not as efficient, but it can still make ATP when there isn't oxygen. We have a video about that. We can't emphasize enough how important the process of making ATP is for cells. For example, cyanide, which is found in some rat poisons, can block a step in the electron transport chain, which would block ATP production. A poison that prevents ATP from being produced can be deadly. With the important role that mitochondria have in ATP production, there is also a demand for increased research on mitochondrial diseases. We are confident that the understanding of how to treat these diseases will continue to improve as more people like you ask questions. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, again, I like that video just because it gives you a good visual of all 3 steps of cellular respiration, as well as, the inputs at the beginning and the outputs at the end. So here's our review question to end this process. So in cellular respiration, glucose is broken down to produce the end products, A, NADH, water, and oxygen. B, NADH, FADH2, and water. C, ATP energy, water and carbon dioxide. Or D, ATP energy, water and oxygen. So what are the end products of glucose being broken down in cellular respiration? If you chose C, I would agree with you. So remember, glucose and actually oxygen are the inputs of cellular respiration, and then the end products of this process include ATP, 6 water molecules, and 6 carbon dioxide. All right, so by now you should be able to describe the relationships of glycolysis, the Krebs cycle, and electron transport chain. So basically the inputs and outputs of those, identify the amounts of NADH, FADH2, and ATP produced during cellular respiration, as well as state that overall chemical equation for cellular respiration. So just like the Amoeba Sisters video alluded to at the very end, what happens if we don't have oxygen present? So the final video in topic 4, we're going to look at what is fermentation.